So our third macromolecule are proteins. Proteins are probably the most diverse of the four macromolecules. Well, I guess nucleic acids can also be diverse in their sequence and then meaning, and they're connected to each other. But as I said before, your cells can produce about 80,000 unique proteins. And this is because there are more monomers of proteins. Those are amino acids. There's more variety in the monomers. And then the sequence of the amino acids makes the proteins have distinct functions. And this results in them being the really molecular machines that take care of all the activities of your cell. And so they have a lot of diverse functions, as I'll go through in just a moment. They are abundant in cells, and they do all kinds of different things in cells based on their different structures. So this is just some categories of proteins and what they do. As we'll talk quite a bit about more later on in the semester, proteins act as enzymes, which means they catalyze chemical reactions in your cells. We've been talking quite a lot about antibodies and whether you have antibodies to a specific virus. Antibodies are protein-based compounds that cells make and secrete outside the cell to attach to foreign invaders like viruses. Some proteins just store up amino acids for nutritional purposes, like albumin in an egg. There are proteins embedded in your cell membranes that allow materials to cross those membranes. We'll talk more about that this unit. And then proteins include things like insulin that gets secreted into the bloodstream and affects your metabolic processes in your cells. There are proteins involved in communication between neurons and other cells. We call these neurotransmitters. Some of those are proteins. We have proteins that make up our muscle. You may know that if you want to have a good supply of protein in your diet, you should have a lot of meat in your diet because meat is abundant in protein. Dairy products also have a lot of protein in them. The muscle is largely protein-based, and the proteins in the muscle that cause the muscle to contract are shown here. And then we have proteins that give our skin its flexibility and support. So collagen and fibronectin. Collagen is probably one of the most abundant proteins in your body that affects your skin, the structure of your skin. So this is just some examples. We'll be coming back to many examples of proteins as we learn about them throughout the semester. The monomer protein is an amino acid. We mentioned this in the last unit. It has an amino group and a carboxyl group with an asymmetric central carbon. And then all proteins have some sort of side chain on them. Think of this as just a generic here, but I'll show you the different side chains on the next slide. But this is the basic function of all amino acids. Here are some side chains highlighted in yellow, and they also then can have functional groups associated with them. And these functional groups have different chemical properties that puts the proteins into these different groups. So everything on this slide is considered a hydrophobic amino acid based on the hydrophobic side chains that are contained in those amino acids. So if you look here, all of these highlighted in yellow are hydrophobic. If we look at the backbone of the protein, notice they all have an amino group, a central carbon with a hydrogen attached, and a carboxyl group. So that's inside the blue box for each of these. These are polar amino acids, so they have polar functional groups associated with them, like these hydroxyl groups here, the amino groups here, sulfhydryl group here. So these amino acids are hydrophilic amino acids. And then these amino acids are either acidic because of the carboxyl groups or basic because of the amino groups attached to them. So I won't ask you to memorize and name, be able to identify lysine, but if I showed you something like arginine, you should recognize the amino groups here and be able to predict that's going to be a basic amino acid. Proteins get put together by dehydration, just like the other polymers do, into a polypeptide, a big long chain of amino acids joined together. And the bond that connects them is called a peptide bond. And then remember that the hydrolysis reaction is breaking that bond. That's what happens in your stomach and small intestine when you digest proteins. The other thing you should know is a polypeptide has a distinct amino terminus, amino end, and a carboxyl terminus, because a peptide bond always forms between the carboxyl group of the first amino acid and the amino group of the second amino acid. And so the free end of a protein is going to be an amino group, and then the beginning end, I should say, and the final end is going to be the carboxyl group. So the amino group is sort of your beginning of the protein. The carboxyl group is the end of the polypeptide. Proteins have 4 levels of structure. The first level of structure is just the sequence of amino acids. Because of the number of different amino acids, at each position on the chain, you can have a different amino acid. So the varieties here is basically infinite. And the sequence of a protein, the primary sequence or structure of a protein is the primary structure is the sequence of amino acids from 1 to however many amino acids are found there. And again, remember the beginning is the amino end. So here we see the amino acid backbone with the amino group, central carbon, carboxyl group, amino group, central carbon, carboxyl group. And we don't have any, these are just generic. They haven't added the side chains on these. The second level of structure of a protein, how it folds up on itself to form a 3-dimensional shape, is called secondary structure. And there are 2 key types of secondary structure: alpha helix, where hydrogen bonds between the amino acid backbones form this helical structure, or this pleated structure called the beta pleated sheet. Those are both types of secondary structure, and they form due to hydrogen bonding between the amino acid backbones. Tertiary structure happens when the side chains of amino acids interact with one another, and they can interact with hydrophobic interactions, where 2 hydrophobic amino acids can get together and cluster together. They can form ionic bonds, where you get an acidic and basic amino acid finding each other. They can form hydrogen bonds, and they can form those disulfide bridges between the sulfhydryl groups found on a couple of amino acids. These types of interactions then help to stabilize the 3D shape of a protein, and this is known as tertiary structure. Some, but not all, proteins also have quaternary structure, and this is where 2 or more amino acids, 2 or more polypeptide chains come together to form 3-dimensional shape. So this particular protein is hemoglobin. In it, there are 4 subunits here, or 4 independent polypeptides that come together to form the hemoglobin protein. And so this protein then is functional when all of these 4 subunits have joined together to make hemoglobin. So not all proteins have quaternary structure, but if they have a protein partner that they have to get together with to function, then they have also quaternary structure. Here's collagen, that really abundant protein I told you about. It's made up of these 3 polypeptides that wrap around each other to form this rope-like quaternary structure. And there are a variety of ways to represent protein shapes. So we've seen models, molecular models already in this class. Here's a 3D or space filling model. This ribbon model is pretty common, where you get sort of like the skeleton view. You take out the surface view and instead just see the skeleton view, where you can see those aspects of alpha helix and beta pleated sheet in here. That's a fairly common way. And then this wireframe model gives you a little bit more detail than the ribbon model does. And then in our textbook, we sometimes model these in a much more cartoonish form, where we're really showing something that has this complex 3D shape into basically just a blob with some structures associated with it. So you'll often see that as well. Here's an indication of how important shape is in the stabilization of these 3D shapes in the function of a protein. This is antibodies. The blue and the red are antibody proteins. And I hope you notice how their shape, especially at this margin here, matches up with the shape of the flu virus. And so this is how the antibodies recognize that virus, by matching the shape of the antibody with the protein on the virus itself. So shape matters in protein function. In all levels of biology, shape matters, but for protein function, shape really matters. So here's an example of what happens when you're with protein shape, or sorry, this is, I guess, just a summary of those levels of structure. Primary structure, sequence of amino acids. Secondary structure, hydrogen bonding that forms alpha helix or beta pleated sheets. Tertiary structure, interactions between the amino acid side chains, including ionic bonds, hydrophobic interactions, hydrogen bonds, and disulfide bridges. And then quaternary structure, where two or more proteins come together. And here's our example of the importance of all these levels of protein structure. So here's the sequence of hemoglobin, that protein that carries oxygen in your blood. Here's a change in protein in the primary sequence or primary structure of hemoglobin, where you get a substitution of one amino acid for another. So you have glutamate here and valine here. That changes the primary structure of this protein. These two amino acids are different from each other. Glutamate is an acidic amino acid, so it's going to be found typically on the surface of a protein, interacting with the cytoplasm, other polar substances on the surface of the protein. Valine is a hydrophobic amino acid, so it's not going to be hanging out on the surface of the protein interacting with water. It's going to hide inside the protein away from water. And that causes this protein, when it folds, to change its shape. So normally the hemoglobin is going to have this sort of round shape, and the valine, because it pulls those amino acids into the interior of the protein to get away from the water in the cytoplasm, is going to change the 3D shape of the protein that's produced. And that in turn changes the way that the hemoglobin subunit associates with its protein partners. So here's the hemoglobin with the 4 subunits, and here's the abnormal form with those abnormally shaped beta subunits. And what happens is those abnormal hemoglobins then aggregate with one another and form these big long chains. And that aggregation changes the shape of the red blood cell and leads to the sickled shape of the red blood cell. So this disease, sickle cell anemia, which is a genetic disease where the genes change this primary structure of a protein, results in a change in the shape of the red blood cell due to a change in the shape of the protein that's produced by that genetic sequence. Proteins can denature, that is, unfold from their natural 3D conformation. Factors that affect denaturation include heat, acid, salts. All can cause proteins to denature, and this is usually relatively irreversible. So like if you heat an egg white, it's going to turn white. If you leave it sitting on your stove and let it cool down or put it in the fridge, it's not going to go back to that clear color, right? Because that denaturation was so severe that it can't refold back into its normal shape.